Hello, this is Nick Ruszala. I'm going to be doing a presentation on mitosis. Alright, so mitosis has a bunch of different phases, but for this I'm going to focus on the M phase and the different steps of that, which is prophase, prometaphase, metaphase, anaphase, and telophase. Alright, let's start off with prophase. So in prophase, basically the characteristic of that is that your nuclear membrane is present, but then it starts to break down. And so basically what's going to happen is this nuclear membrane is going to break down, allowing these chromosomes to get free and start moving around the cell. And the spindle apparatus, which is right here, is going to start to form, and it'll become present, and it's going to start forming around the cell to form a sort of a skeleton, if you will. Okay. And so next, chromosomes. These chromosomes are going to start to condense and become visible. They're not fully condensed, but they'll be starting to condense. At the end of prophase, they'll be fully condensed. And then chromosomes here appear as two sister chromatids, and they're held together by a centromere in the middle of them. Alright, so next we're going to move on to prometaphase. So basically what happened is the nuclear membrane broke down, these chromosomes became loose, spindle apparatus forms. So now the nuclear membrane is absent, broken down, fragmented. Alright, and the spindle apparatus is assembled, formed, and chromosomes are condensed completely. Each of these chromosomes is now going to be positioned so that the kinetochores of these sister chromatids are going to be attached to the microtubules of this spindle apparatus so they can move around. Oh, sorry about that. And so basically what's going to happen is these chromosomes are going to start attaching to the spindle apparatus. And they have to be positioned in a way so that each of these sister chromatids, which is like this little section right here, each of these sister chromatids has to face a different pole of the cell. Each of these has to face a different pole, and they'll be attaching to the spindle apparatus, and they're going to start moving towards the center of the cell so they can line up equally along the middle. Alright, so next there's the stage is metaphase, which is basically a single point in the process. It's like a split second. It's like just an event that happens. All of the chromosomes are lined up along the metaphasal plate, which is also like the center, the equator of the plate. These chromosomes are positioned, each of them's at a different, each of these chromatids are pointed, each of these chromosomes are pointed at different poles of the cell. Our nuclear membrane it's still gone, it's still absent, broken down, fragmented. Our spindle apparatus is formed yet again. And basically what's going to happen is the spindle apparatus is going to start creating some sort of tension because now these are at the middle and they're going to want to start to pull apart. And that's when we get into anaphase. Basically, the anaphase, nuclear membrane, still broken down, fragmented, but our spindle apparatus is present. But now, at this time, the poles of the apparatus are pulling each other apart. And this pulling apart takes these chromosomes, which are still condensed, and they're going to be the proteins holding these two chromosomes together, like the two chromatids together, are going to be degraded, and these sister chromatids are going to split. And once they split, they each become their own new chromosome. So each of these poles is going to have 4 new chromosomes moving towards the end of the poles by the spindle apparatus, which is pulling itself apart, basically. Alright, which is going to lead us finally to telophase. And basically, telophase, you're going to have a nuclear membrane is now going to start to reform around the chromosomes, and our spindle apparatus is going to be present, but it's going to be disassembling. It's going to be breaking down, getting smaller. And our chromosomes, and now each of these four new chromosomes, keep clicking. These 4 new chromosomes are going to be, they basically migrate to the poles, and they're going to be clustered at these poles. And what's going to happen is at the poles, the nuclear membrane is going to start to form around these new chromosomes so that 2 new cells start to form. And basically what's going to happen is these two new cells are going to have the same number of chromosomes as our parent cell. So this one has 4 chromosomes right here, and now there's 4 here, 4 here. All right. And after that, there's going to be cytokinesis where the cell's actually going to split and they'll have 2 daughter cells. But we're not worrying about that in this presentation. All that we're worrying about is just prophase, prometaphase, metaphase, anaphase, and telophase. All right. I believe that is all. Thank you all for listening.